Hi students, in this video I am going to explain about the explanation of the concept of the dominancy. Generally majority of the students or people used to get the doubt in this one, how it is forming, mainly where in the no enzyme products to produce gene or non-functional enzyme produce gene. What is this? And how the residue used to form, all the things I am going to explain you in this. And diagrammatically also I am going to explain in the codominace also in the same video. It is most important for the 2026 also, remember it, because they didn't ask the question in this one, but the chances are there to ask. And now whenever we are seeing this one, the copies of gene or pair of allele contain information of gene expression. Any expression will be followed by the gene only. Genes will be in the pair in the diploid organisms. And the diploid organism, the pair is also called as allele. And the allele both together used to express the character. It may be the homozygous or heterozygous condition. Remember it. Now, one of them may be different due to some changes that it has undergone. When it is a heterozygous condition, both the alleles of the two, one of the pair two are there. One may gone some change in their structural change that leads some expression difference. Expression difference also will be shown. That expression difference we used to see in the F1 generation according to Mendel. Means F1 generation always used to resembles the parents, one of the parents. All these things will come in this one, and how the residue used to form also I am going to explain. Now, this leads changes expression of that modified allele that I already told, that is the heterozygous condition. It used to give the expression. Not only in the heterozygous condition, dominancy will be expressed. In the homozygous condition, the gene which is modified, that also will be expressed. For example, sharp darkness. That is the thing. So, for example, the allele forms enzyme for transformation of S, means substrate. Here, what it is to express, the gene is to express its character in the form of the production of the enzyme, which is the substrate will be converted into the product. And in this one, what they will be produce enzyme for the expression of the character. That is the transcription and translation what you are going to read in the molecular basis of inheritance. Now you see here, for example, allele forms enzyme to transformation of substrate as in the three characters. Now you see here, first one, the normal or less efficient enzyme. Means the enzyme will be expressed its character by producing the gene will express its character by producing the enzyme. That enzyme may be normal. Normally it is producing means a tallness, or it is a less. If it is producing less, it is a shortness, like that we can understand. So the both will be the functional. Both the allele which is the modified and unmodified, both will be expressed in the first case. Remember it, this is the first case. Or a non-functional enzyme. What is this non-functional? Means enzyme is functional, but it is non-functional enzyme. What is this? Remember it. The enzyme will work based on its active sites. The enzyme is there, but active sites are default, or they may be not formed. Then the enzyme will be appeared. What is the enzyme? Tertiary structure, already studied. that hollow wool ball, all the things. So in that one, because of some modification, what happens? It is not having the active sites. Then it is non-functional. Understand? And next, one more third point is no enzyme at all. The enzyme, whenever the transcription is going on, the second or third triplet, maybe the stop codon, it might have become stop codon. So it cannot transcription won't be taken, translation won't be taken. But it is present in the allele, the pair of allele. So it is non-functional because of the not having proper active sites, or transformation failed, transcription is failed because of some, for example, I am telling stop codon modified in the third triplet, over it stops, but it will work because second allele is functional. That only is going to tell. The example can I tell? That is the O group blood. O group blood, it is not having I, that I, my allele won't produce the sugars. Why? Because it is not having that sugar producing enzyme. That is the second and third, remember it. That is the second and third. Understand, no? So non-functional or no enzyme. Best example is the red blood cells. That is the O group. If it is, then it is recessive. Whenever it is with the A, it is forming the A group. By the I gene is it is the recessive. You are studied now? That is the thing. Understand it. Try to understand. Now, in first case, the modified allele is equivalent to unmodified allele. It will produce the same phenotype, trait, and functional. That also it already told. What is the first case? The normal or less efficient enzyme. The trait is tallness. For example, tallness normal, if it is producing normal enzyme, then tallness. Modified and less enzyme producing darkness. Like that, if you think, then you can understand very easily. Now you see here. So it will produce the same phenotype, trait, and functional. So functional means it is appearing height, either it may be tall or short. For that only I am telling you the examples. It will be easy to you. Now we'll see. So phenotype, trait. This type of pair are very common in diploid organisms. This is the most important point. Means here non-functional enzyme or no enzyme, it is very less places only it will be taken place. Most of the cases, the allele may be modified, but it in the expression condition. That is the most important. Means people will get the doubt here only. What is this one? Majority of the genes will be expressed in the normal or less efficient enzyme production only, but very few. That's why he is telling to you to understand the ratio, how it is developing. So the ratio in the second and third, but whereas second and third are present in the allele, phenotype, trait depends on the functional allele for expression. For example, here the capital T is there, small t not produced. Just for example, I am telling. And but it will be expressed or not. One of the allele is functional, so it will express. Either maybe tall or short, or the blood group. If you see A, B, if it is homozygous O, that is the condition. Understand, no? So it's depend. If it is not having the non-functional enzyme, second allele, of that pair, one allele is a non-functional producing enzyme, it is non-functional, or it is not having the no enzyme is produced, but it is having the other allele that is expressed. Now here one more twist is there. The recessive character which we can be expressed, that is the first case, will become dominant when? Will become dominant. Because the shortness, for example, if you take, shortness is the recessive. But one of the character, one of the allele is short. And second allele is non-functional or no enzyme. Then shortness will be this will express it or not. So single allele shortness also will be expressed. Remember it. Why I am telling in this way? He can create the question in the need. So try to understand. So it depends upon the functional allele. When the second allele becomes non-functional or no enzyme, it is producing. In this way, the first maybe dominant allele or recessive allele, whatever it may be, they are the functional. But second allele is not completely non-functional or no enzyme. Then it will work or not? It will work based on functional allele. But when second and third present in the allele, phenotype trait depends upon the functional allele for expression. Understand, no? Now, in this case, the functional allele becomes dominant. Clearly, he has given. Means if it is the shortness is there, it will be exhibited even though second allele is not there. When he will ask. That is when it will be the second allele is not producing the enzyme or no non-functional enzyme. I think you might have understand properly. Now whatever the modified non-functional and no enzyme, those place slowly it is called as a recessive. Recessive has developed like this. Now we come to this non-functional and recessive. It can be seen in the blood group O. O blood group is O. It is that is the I, small I, in the blood group formation. It is not having the sugars on the surface of the plasma membrane. Why? Because sugar producing enzymes are not produced because of that enzyme required for that one is modified. Means as a non-functional or no enzyme. Now understand, no? That is the main thing. He can ask in that way also. Remember it. So this is about the dominancy. Dominancy character will develop in the means which will be expressed even though in the heterozygous condition also, that is F1 generation mainly, that is called as dominant character. That dominant character expression, how it will come, that for example he has given 3 steps. First one is normal or now you see the normal or less efficient enzyme, or this is the common, means most of the things used to have, and non-functional enzyme or no enzyme produced because of the modification of the second allele. That how it is I explained, but most of the cases it will follow the first one. That is the thing. Now we'll see codominancy. Codominancy here, the thing is, before knowing the codominancy, what is dominancy and what is the incomplete dominancy? Dominancy means already we explained how it is formed. Means one of the allele will be dominant, means it will be expressed, and the other thing is not expressed, that is the dominant. That is the thing. The complete dominancy F1 generation resembles one of the parent, and incomplete dominancy, it is between. That is the dog flower. Red and white flower gives the pink flower, incomplete dominancy. So dominancy or incomplete dominancy, you came to know already. Now he is going to tell you about the codominancy. Means here in the F1 generation, both the allele used to express its character. That is codominancy. Both the alleles, a pair of allele, both the allele will be expressed. Both will be by pair of allele will be expressed, and that is called as a codominancy. Now you see here, in codominancy, F1 resembles both the parents like in ABO blood group by having three alleles. This is also most important. That ABO blood groups already everyone knows. This used to form by the having three alleles. Generally, the allele will be the pair, but here three alleles used to present. Three alleles cannot be expressed in one organism. Why? Because the organism is diploid. Only two genes can be there, will be there. So, if you want to know all the genes, but it is having the more than two alleles, you have to study the population. Means in different will be there. Means here some people are having A group, B group, O group, but and different formations will be done. When you will come to know, if you check the different people in the population, then only you will come to know. That only he has explained there. Now the complete dominancy F1 generation resemble one of the parent, and the incomplete dominancy, it is not in between. That is the dog flower. Now you see here, in codominancy, F1 resembles both the parents like in ABO blood group by having three alleles. More than two alleles are present in that trait or the gene or expression. In that one, they are the, what are they? Three alleles. You see here, he has given. That is IA, IB, I. The gene used to have three alleles as a IA, IB, and a small I. So, produce significantly different sugars by the blood. IA is also called as multiple allele. This is most important. Means allele generally must be in the pair. If it is more than pair, then it is called as a, we are calling it as a multiple allele. And IA means A and B produces slightly different sugars in the red blood cells. Why the A and B are forming means in the sugars produced, the sugars polymer will be produced by those cells, red blood cells, and they will be protrude, means they will come out. That he has given clearly in the plasma membrane structure. Go and see. Sugar chains used to present on the plasma membrane. So here the thing is, the sugars which are produced by the cell, red blood cell, they will be slightly differ. That is the polymer of the sugar, and they will protrude. They used to come out from the plasma membrane, and it used to, the difference used to make them as A and B. The difference has only become the A and B, whereas what I is there, no, that is third allele. It won't produce any sugar because of non-functional enzyme. It may produce, or it is not producing no enzyme. That is because, why I am telling, that is non-functional enzyme only, but some references has given some difference there for that one. Take it granted non-functional both. The I is having the gene or not means I is having the participating in the allele, that is also having the gene, non-functional, or no enzyme producing. That is the thing. So produces slightly different by the blood cells, and sugars protrude on the surface of the plasma membrane. That you see the plasma membrane structure in the 11th class. I allele does not produce any sugar because it is not having the non-functional, means enzyme will be produced by the gene only, or no enzyme will be produced. Enzyme, why the enzyme is required? To form the sugar. The sugars are also two types. The slight difference will be there in the A and B. That only is telling, nothing. And A and B, that is the IA and IB, are the completely dominant than the I in heterozygous condition. Whenever the heterozygous condition is forming with A or B, A may form heterozygous condition with I, means O, or B may form heterozygous condition with O. In that condition, A, whenever it is A will be the dominant, and B will be the dominant than the O, I, that is the. So here, both A and B are showing the dominancy with I, that is the O group. But whenever the heterozygous condition forms in between A and B, they will show the codominace. A will produce according to its sugar it will produce. B also will produce according to. So the parents are the A and B. AB group came out. The AB group used to have the both the two types of the sugars which are slightly different for the formation of the A and B. We is told these things will present in the AB group. That only is telling. A and B are completely dominant than I in heterozygous. In homozygous condition, A and B I forms a B group. If it is A with A, it will be homozygous, A group it will be. That I shown in the chart play. That I will discuss that also. A and B blood groups, A and B heterozygous condition codominant. So if the A and B both are forming in the allele, in that one codominance means A will produce blood group cells, it will produce that type of the sugar, B will produce such type of sugars. So both will present. For that only, that is the universal acceptor we used to say, and universal donor is the O. That a zoology person can discuss all the things. Now, just I prepared some chart type for better explanation. How many phenotypes and genotypes used to come from this? So here A, B, O. That is the IA, IB, and I, small I. Here A may be with homozygous with AA, or A may be by heterozygous with O, that is I. So it is then the A will be the dominant, because if the heterozygous A is the dominant, homozygous also A will be there. So A can form homozygous A can form the dominant, means by showing the dominant character A, it can show with O, heterozygous condition. And now B also same. It may be in the heterozygous condition with the I means O group, and heterozygous condition dominance of the B. B will be expressed because O is not having the sugar. That is the thing. But whenever A and B are forming, then this is the codominancy will be seen. This heterozygous condition A and B are present in the alleles, then it will be codominancy. Dominancy, codominancy. Both he explained in this. I think if you write this one, you can easily read the thing. For example, here, how many genotypes are there? See, A and B and O. These are the three. And B with heterozygous, A with heterozygous, that is the 5 and 6. And A with O, heterozygous. So now I given the number 1, 2, 3, 4, 5, 6. Because in the chart, in the diagram, he has given in the textbook, 7 are there, 2 are the AB. So they counted 5. But whenever you are seeing the phenotype, how many? Because phenotype, whenever we are seeing, this is the phenotype is different. Just one, if you remove, that is the phenotype, it will come. How? This is the phenotype, and A also phenotype will come, B also phenotype will come, O will be there. So totally phenotype also will come. Already he has given in the textbook. This many phenotypes are there, how many genotypes are there. It is very easy to find out. Is any difficulty? Not difficult, I think. Now you see here, and one more he has given, that is a pleiotropy. Pleiotropy directly not mentioned, but he mentioned at the end of the chapter. That pleiotropy, it is very simple topic, but some people are getting lot of doubt in this. For that I prepared this chart. Now what is the pleiotropy? One now this is this is what we are seeing in the. It is ABO blood groups are 3 alleles. It is also called a multiple allele. Now alleles may be 3, but a pair of allele may be expressed in, it may express 2 characters. 3 pair may be express 1 character by the having 2 only, because in the diploid it is. But at the same time, one gene pair may express more than 1 character or the trait or the trait. That is, you can see in the pear plant seeds. That is the see here. A pear seeds single gene produces more effects. Effects means characters. Single pair only will give more effects. How the effects are, and that effects also, one is the dominant effect, one is the incomplete dominant effect. One is the dominant effect, one is the incomplete dominant effect. Both are present in the pear seeds. This also need important question. Now you see, here I given it a different colors. You see, try to understand. If this pear seed, that it is having the capital BB and capital small bb. Capital BB, whatever it is there, that is the homozygous condition. It is to have round and large starch grains. Because of the starch grains are big, the seed become round. That is the thing, nothing. And in the small bb, which is the recessive, that is a wrinkled and a small starch grains. Because of small starch grains only, it is not fully expand the seed will become wrinkled. Now it is okay. Now whenever we are crossing this one, means if F1 generation, capital B, small b will to come heterozygous condition. Here only the problem will come. That is, capital B is dominant than the small b. So round will come. Dominancy will be observed in the capital B. Capital B is dominant than the small b. So the seeds are round, same color I given. But the seed starch grains are small, starch grains are the large size. Intermediate, that one I given some other color. Intermediate starch grains are coming. So intermediate starch grain production is, it is the, we used to say this one as the incomplete dominancy. Means dominant gas is having large starch grains and recessive is having small starch grains. But whenever the F1 generation we are observing, that shape and shape of the seed and starch grain size will be determined by single gene, that is in the pear seed. Out of these, two characters are maintained by the single gene, even though the two characters used to differ in the dominancy. In one character, they will act as dominancy, and the second character it will work as the incomplete dominancy. That is the thing. It is very easy. So intermediate starch grains, capital N. Therefore, and he has given one conclusion. Many students are asked, "What is this, sir? What is this paragraph he has given?" The thing is, here, by seeing the codominancy in the in the blood group and in the pear seeds, dominancy is not a autonomous character, because if it is the dominant here, it is dominant than the O group there. Here B is dominant than the B. But in the expression of the second character, it failed. It become incomplete dominancy. So that only he given. Dominancy is not a, it is a dominancy is not a autonomous feature. In single gene produces more efforts. The efforts dominancy also may be different. Understand, no? This is very simple topic actually, but the students used to get the so many doubts. I am expecting that he will catch in 2026 or in the, I think one bit he can catch beautiful bit, because only he is asking if the parent is the group A, that is the blood group is A, father, mother is the B, and the children are the O. How it is possible? Very simple. That is the both are the heterozygous. So heterozygous means AO and BO. O will come. Such type of simple questions here, but chances are that he can ask more questions in this one. So if you like this video, please give the like mark and subscribe the channel to know more things like this in my channel. Thank you.